Good morning, everyone. Thank you so much for coming to these D2K presentations. My name is Andrew Warburn. I'm a second-year medical student here at the Icahn School of Medicine at Mount Sinai. I'm very excited to talk about my project, D2S2, that I had the privilege to work on over the summer with the Ion Lab for the last 10 years. There we go. Perfect. So before I sort of dive into the project, some background on connectivity mapping specifically. So connectivity mapping, as some of you might know, is the principle that there can exist a data set full of drug-induced gene expressions that a user can then take a signature of interest and then see if this drug mimics or reverses that signature. Currently, the L1000 is the largest connectivity map and most comprehensive, with over 1.3 million samples, over 400,000 signatures across 978 landmark mRNAs. The L1000 has been absolutely vital in drug discovery because you don't need to know the activity of the drugs in order to, or you don't need to know the drug target to predict the activity. However, as good as this may sound, there's a lot of skepticism and criticism around the L1000 because all the signatures are based on cell lines rather than an entire organism, and the reliability of these low-cost transcriptomics have yet to be completely validated. So this brings me to the purpose of this project, D2S2, which is a web application that aims to globally validate the L1000 database against clinical applications seen in the Mount Sinai electronic medical record, or the EMR. And not only will it take the EMR data, it'll also look at different gene signature sets from other databases such as CREDS, GeneShot, Enricher, X2K, GWAS, thank you, Allison, and the UK Biobank. And so the aim, ultimately, of this application is to explore the consistency between the L1000 signatures and the FDA-approved drugs and the disease signatures for these diseases that these drugs are used to treat based on the EMR. So here's an overall of the methods of how the application sort of takes from each different data set. So the user can either query a drug or a disease of interest. And so, for example, if you wanted to look up warfarin, you would go into this drug pipeline here. You get L1000 FWD signature. You also get a CREDS disease signature as well. You'll pipe those signatures through an X2K to get some transcription factors and kinases. And then it'll also do what's called a reverse query through the Sinai EMR, where we basically match a drug to the diseases that it's most commonly prescribed for. And so those diseases, usually the top 3, will be sent through the disease signature pipeline, wherein it'll also go through the CREDS disease signature platforms, the Enricher platforms, and then it'll also go through the X2K to see the similarities between those transcription factors. We'll also get other signatures from GeneShot, UK Biobank, and GWAS as well, so we can compare globally across all of these different data sets. And then we can see the drug and disease signature comparisons from that. So here's an example of what the EMR, sort of a reduced look at the EMR looks like. So again, with the warfarin example, you can query warfarin, and we can see the number of different occurrences and use cases for warfarin. So we take the top 3, which is atrial fibrillation, congestive heart failure, and unspecified hypertension. And we take the top 3 because those are the primary use cases for the drug of interest, right? You don't, if you take all of the uses of warfarin, a lot of the times it's a secondary application of the drug, right? You don't prescribe warfarin for asthma or different allergies of types. And so we wanted to make sure that we're taking the drug for the specific use case from the EMR. And so this is an example of what the website will look like. So thank you so much to Shirley for working really hard and making it look really good. And here's sort of the landing search results page. So each of these tiles represents a different database that you're going to be able to collect a signature from. So users are able to download any of the signatures that they select. They can go and select these, visualize the signatures using a clustergrammer, so thank you, Damon, for that. And then from that, you can also identify different consensus lists between these gene signature sets. And then you can also further identify pathways, targets, and mechanisms for drugs, or drugs used in different disease forms. So here's an example of that visualization power that we have through the D2S2 website. So here's 4 different databases: Drug Matrix, CREDS, L1000, and GeneShot. And you can compare the similarities between them. So a lot of the same gene sets from the same places will have similar indices to each other, but you can also see how they're similar against different matrix or different data sources. But besides from comparing different data sources and different signatures from different data sources, you can also see with D2S2 whether or not a drug reverses or mimics a disease signature, which is one of the critical components of a connectivity map. And so here, what we have are the top diseases that warfarin was used for and the signature for reversing or mimicking it. And so this heat map right now is organized by which genes warfarin seems to downregulate. And so you look at the top left, that dark blue, so you can see that warfarin will downregulate genes that diamond blackfan anemia seems to upregulate. So you can see that warfarin has this reversal or predictive of reversal effect on the diamond blackfan anemia. And this can be used, hopefully you can see this power of being able to globally scan this with a drug of interest. You can see that it can not only be used for future drug discovery, but also drug repurposing. So taking currently existing FDA-approved drugs and then seeing new and novel applications to see what they can reverse, or even checking to see if they mimic any disease forms. So currently we have tested 309 FDA-approved drugs from creeds into the D2S2 interface. And we have 828 diseases from creeds that we can analyze, and then we have 266 unique matches from those diseases to the EMR diseases, and then of the drugs that we've mentioned, 309, we have 200 unique drugs that we compare through EMR drugs as well, and with their aforementioned disease matches. So in the future, we hope to continue to improve visualization, continue to map these drugs with their correct EMR usage, and then really see and be able to dissect whether these drugs in clinic are actually reversing or mimicking the disease states and disease forms that they're being clinically used for. Some challenges that existed through this was a lot of the mapping, a lot of the disease states between creeds and the EMR had to be mapped via ICD-9 codes because the ontology wasn't very good, and a lot of the drugs and disease side effects also had to be done manually by yours truly. And so, but overall, it was very exciting, and I want to really thank everyone in the lab for all their help and support. Thank you, Shirley, again, for all of the help and UI and patience with all my buggy code. Thank you, Daniel's not here, but I would like to thank him for fixing the buggy code. And then thank you to everyone else in the lab, especially Avi and Sherry for dealing with me. And yeah, and all you guys for being here and listening to this presentation. Thank you.